morning. Welcome to Lezend online classes. Myself, Dr. Achaya Nalamada. I am going to discuss food microbiology as part of FSO. Right. Well, so there are some organisms that are pathogenic. So these pathogenic organisms usually they cause certain diseases. Yes, they cause spoilage of food material. So these organisms are mostly bacteria, fungi, molds, yeast and virus. So these organisms usually they deteriorate the food quality, so that always they affect the quality of the food, right? As a result of that, they induce certain undesirable changes in the food material. So basically organisms are categorized into two types. One is prokaryotic organism and eukaryotic organisms. Basis of the organization of the cells are categorized into two types: prokaryotic organisms and eukaryotic organisms. In prokaryotic organisms usually lacks nuclear membrane. So genetic material is present, right? Genetic material is present both in eukaryotic and prokaryotic organisms. In case of prokaryotes, nuclear envelope is absent. Therefore, genetic material is simply said to be naked. Whereas in case of eukaryotic organisms, genetic material is surrounded by nuclear membrane. Moreover, the cell has been compartmentalized in case of eukaryotic organisms. Cell organelles are also present in eukaryotic organisms. So, in addition to this, broadly the entire organisms are classified into plants, animals and microorganisms. So, these prokaryotic organisms includes bacteria, blue-green algae, right? The leftover, whatever the leftover is, all fungi and other organisms, so protozoa onwards, they come under eukaryotic organisms. So, in case of prokaryotic organisms, cell organelles are absent, not only nuclear membrane. So, genetic material is present. This genetic material regulates the activities of the organisms, so that this genetic material is always protected by means of nuclear envelope or nuclear membrane in organisms. When it comes to prokaryotic organisms, nuclear membrane is absent. Simply, genetic material is present in a naked form. There is no nuclear envelope around the genetic material. Therefore, it is said to be naked genetic material. Right? This is the case with prokaryotic organisms. So, in case of eukaryotic organisms, the cell itself has been again compartmentalized. The cell has been compartmentalized into many cell organelles like nucleus, mitochondria, Golgi complex, endoplasmic reticulum, right? Nucleus, ribosomes, vesicles, Golgi complex, cytoskeleton, mitochondria, right? So, in case of prokaryotic organisms, so all cell organelles are absent except ribosomes. So, the ribosomes present in prokaryotic organisms is of 70S type. So, except ribosomes, all cell organelles are absent in case of prokaryotic organisms. That includes nucleus, mitochondria, Golgi complex, endoplasmic reticulum, centrioles, lysosomes, right, even microbodies. So, all these cell organelles are absent. Only plasma membrane, that is the protective layer around the cell, this protective barrier is present, genetic material is present without genetic nuclear envelope. In addition to that, finally ribosomes are present. So, these ribosomes are part of protein synthesizing machinery, right. So, right. In case of eukaryotic organisms, the necessity for compartmentalization is to discharge the functions in an efficient manner. For example, the mitochondria is specialized in generation of energy. This is called as powerhouse of the cell. Right? It generates energy, it synthesizes ATP, nucleus stores genetic material, lysosomes stores hydrolytic enzymes that are involved in hydrolysis, ribosomes are involved in protein synthesis, Golgi complex performs glycosylation of material. It attaches carbohydrate moieties either to lipids or to proteins. It is also involved in cargo transportation. So, endoplasmic reticulum is part of protein synthesizing machinery. So, rough endoplasmic reticulum synthesizes proteins, whereas smooth endoplasmic reticulum is involved in lipid biosynthesis. So, the difference between smooth and rough endoplasmic reticulum is presence or absence of ribosomes. When endoplasmic reticulum is embedded with ribosomes, it is said to be rough endoplasmic reticulum since it appears rough under microscope. When the endoplasmic reticulum lacks ribosomes, it appears smooth, therefore it is called as smooth endoplasmic reticulum. So, functionally again they are different. So, lysosomes are usually having a group of an array of hydrolytic enzymes that are involved in hydrolysis of food material. So, comparatively speaking, in prokaryotic organisms, cell organelles are absent. For example, nuclear membrane or nuclear envelope is absent around the genetic material in case of prokaryotic organisms. Nucleolus is also absent. Then genetic material is common, right? Whether it is whatever the number of chromosomes, genetic material is present, but without nuclear envelope, that is in case of prokaryotic organisms, right? In addition to that, there are major differences, right? So, the cell division is always by fission, binary fission in case of prokaryotic organisms, whereas in eukaryotic organisms, mitosis is the common phenomena for cell division. So, there is a difference. When again in prokaryotic organisms, the number of chromosomes are few, genetic material is simple without introns. In case of eukaryotic organisms, the complexity of the genetic material is more. As the size of the organism increases, the necessity for complexity of the organism also increases. So, that genetic material is always complex, more number of chromosomes are present. Again, genetic material is linear in eukaryotic organisms, whereas in prokaryotic organisms it is circular. So, the membrane itself is phospholipid bilayer both in prokaryotic and eukaryotic organism. The plasma membrane itself is composed of phospholipids in two layers, therefore it is called as phospholipid bilayer. In between there are proteins. So, these proteins acts as gateways, receptors. So, there are different functions are attributed for these proteins. So, membrane itself is specialized in many functions like maintenance of selective permeability, it is involved in recognition, transportation, etc. So, but in case of prokaryotic organisms, the membrane is lacking sterol. Cholesterol molecule is usually present in eukaryotic cell membrane. When it comes to plants, when it comes to prokaryotic organisms, so these sterol molecules are absent. Even in plant cell membrane also, cholesterol is absent. Instead of that, there is stigmosterol is commonly present in case of plasma membrane of plant cells. Right. So, endocytosis is usually common in case of eukaryotic organisms, that is engulfment, intake of food material by phagocytosis. Even exocytosis is also a common phenomena, whereas when it comes to prokaryotic organisms, they cannot endocytose. Simply phagocytosis and phagocytosis is common phenomena, right? Phagocytosis is cell drinking, right? Intake of liquid food material, right? So, mitochondria, lysosomes, Golgi complex, endoplasmic reticulum, vacuoles, ribosomes, right? Except ribosomes, all these cell organelles are absent in case of prokaryotic organisms. As I said, mitochondria is involved in ATP synthesis, that is power. that is the energy source for every living organism. So lysosomes stores hydrolytic enzymes. These enzymes are diversified in their functions. Golgi complex is involved in glycosylation, addition of carbohydrate moieties, and even transportation. Endoplasmic reticulum is involved in protein and lipid biosynthesis. Vacuoles are storage in function. So these ribosomes are part of protein synthesis, right? So they are usually embedded on ribosomes, or sometimes they may be in a dissociated form, right? These cell organelles are absent in prokaryotic organisms, whereas in eukaryotes these cell organelles are usually present. Again, the cell is said to be compartmentalized. The interior of the cell itself has been divided into many compartments. Therefore, it is called as compartmentalization. The necessity for compartmentalization is to discharge the functions in an efficient manner. Again, cell wall, this is an extra layer. It is present in prokaryotic organisms. For example, in bacteria, beyond plasma membrane, there is one more extra layer. This is called as cell wall. This cell wall is made of peptidoglycan. That it includes, the cell wall includes, in addition to that, cellulose, hemicellulose, right, peptidoglycan. In case of fungi, a eukaryotic organism, chitin is present, right, whereas in prokaryotic bacterial cells, right, a cell wall is present. This is made of peptidoglycan layer. So this is also having peptide, protein and carbohydrate moiety. Cell wall itself is, right. In case of eukaryotic organisms, in animal cells, cell wall is absent. Right, just they are having only outer protective plasma membrane. So beyond this, there are no external structures. In case of plants, cell wall is present. Right, in plants, cell wall is present. Cellulose is the material present in the cell wall. In addition to this peptidoglycan, when it comes to fungi, right, these fungi may be either unicellular. Molds are a category of fungi that are multicellular. So in case of fungi, chitin is part of the cell wall, right. In case of algae also, cell wall is present, right. So these are eukaryotic organisms. Again, when it comes to locomotion, movement, flagella are present in prokaryotic organisms. In bacteria, flagella are the locomotory organs. In case of animal cells, especially in case of lower organisms like protozoans, flagella is the locomotory organ, or maybe cilia is also a locomotory organ in certain ciliates like protozoan organisms, right. Yes. So bacteria is a prokaryotic organism. So this bacteria has been divided into two categories on the basis of the composition of the cell wall. It is gram negative and gram positive. So coming to this bacteria, these are minute, right. When it comes to the size, they are very minute in size. So usually they range in microns. So from microns to millimeters itself. Theomargarita namibiensis, that was the largest bacteria. Pleuropneumonia like organism, PPLO, is the smallest one when it comes to the size dimension. So these bacteria are, right, usually minute, microscopic. Therefore it requires some microscope like instrumentation for visualization itself, right. So usually they measure from 0.2 to 0.5 microns in diameter, right. So Leeuwenhoek, he was considered as the person who designed lens, right. He was a lens maker. He designed the first simple microscope, right. With the advent of microscope, the bacteriology has been revolutionized, almost the entire microorganisms. So that facilitated invention and advancement in microscopes, revolutionized the entire field of microorganisms, so that it enhances, it enhances the size, magnification phenomena. So enlargement of the original size of the image is called as magnification. You can go on increasing the size of the pathogen by using this microscope. So in addition to that, again one has to consider resolution phenomena. If you go on increasing the magnification number of times, so that it may result in a blurred image. So that in order to avoid this problem, again one has to consider resolution. So resolution is, that is the ability to distinguish two closely located objects as separate entities, because at microscopic level itself they may appear overlapping. So even in spite of their microscopic nature, one can differentiate these two closely located objects and can be visualized as separate entities. That phenomena is called as resolution power. Always the microscope is simply categorized on the basis of magnification and resolution. So on the basis of that, so the microscope character will be decided. Yes, so there are different types of microscope. Anyhow, electron microscope, scanning tunneling electron microscope, these are advanced one that can magnify the bacteria or pathogen many times, whatever the object that is under study, with increased resolution. Yes, so these bacteria are classified into many types on the basis of the shape, right? Cocci is spherical in shape, round. Bacilli are rod shaped bacteria. Cocci is spherical, right? In addition to that, there are some other microscopic pathogenic organisms. So, right, so for example, spirillum, these are spiral shapes. Spirochetes, so these are a category of pathogens. Actinomycetes are also a filamentous bacteria, branched filamentous bacteria. Then mycoplasmas, these mycoplasmas usually they lack cell wall. Yes, these are cocci. Cocci is spherical, round, oval in shape. Sometimes they may be present in two, called as diplococcus. Di means two cocci, or tetrads. They may be 4 in number, tetrad. Sometimes they may be 6 in number, or maybe 8 in number. So this 8, the cluster of bacteria that is having 8 are called as Sarcina. So Staphylococci, right? This is the bacteria, right? This is resembling bunch, grapes like appearance. So either they may be solitary or they may appear in groups. So on the basis of this, they may be diplococcus, tetrad, sarcina, etc. Right? This is about bacilli. So these are rod shaped bacteria. They are also either they may be single or they may be in association. So when two bacilli are in association, it is called as diplobacilli, right, or streptobacilli, depending upon the number of microscopic organisms present in the association, right. So these are Vibrio comma, a comma shaped bacteria. Spirillum, these are spiral in appearance. Spirochetes. In bacteria, cell organelles are absent, genetic material is naked without any nuclear envelope or nuclear membrane. Ribosomes are 70S type. Plasma membrane, a protective layer, is present in the bacteria. Beyond this plasma membrane, there is an additional structure called as a cell wall. This cell wall is protective in nature. Beyond plasma membrane, there is one more protective layer, this cell wall. On the basis of the composition of the cell wall, bacteria has been categorized into two types, gram negative and gram positive. We will discuss later. So that again, on the outermost surface, flagella are present. These flagella are locomotory organs in case of bacteria. So in addition to flagella, a small hair-like structures are present called as villi. So these villi helps in conjugation, yes, sexual reproduction or recombination. So in addition to this, a specialized structure by name fimbriae is also present. This helps in attachment of the bacteria to the substratum. Flagella are locomotory. Villi, this is involved in reproduction, conjugation, transfer of genetic material is usually mediated by these pili structures in case of conjugation. So finally, fimbriae meant for attachment. Usually they attach the bacteria to the substratum, whether it is in streaming water or some other. So above all, there is one more structure, capsule. This is extracellular material. This extracellular capsule is composed of either polysaccharide or a polypeptide material. So there are inclusion bodies inside the cell. They store some extra material, right? Then there are extra chromosomal genetic material is present. These are autonomously replicating components. They are called as plasmids. So depending upon the number of plasmids,